So, hi everybody. My name is Sally Ann Williams. I'm the CEO of Cicada Innovations, and I'm absolutely thrilled to be here today. We have a special guest, Cyriac Roeding, who we're going to talk to about his phenomenal entrepreneurial journey, the work that he's doing now, and also get some tips and tricks about how to actually bring a really big, bold vision to life. Cyriac has an incredibly impressive resume. I'm just going to touch on a few things because I think it's worth highlighting. He's both an entrepreneur and an investor and has worked on huge companies, global companies in the consumer space, working at the intersection of physical, engineering, and digital worlds. He's previously built at least 3 companies, invested in more than 20, and also been involved in venture funds, including 16Z, Bond, Founders Fund, Greylock, IVP, so many more. He's got an incredibly impressive resume. But I think what's really exciting for me is to have the conversation with him about pivoting away from working on consumer products and building things that you and I all touch and see to his new biotech company, Earli. And Earli's doing something amazing, which I'm not going to steal his thunder on telling us about, because I really love hearing from founders in their own words about why they're doing what they're doing and the impact of it, but working in a completely different space. So Cyriac, thank you so much for joining us today. Thank you for your generosity in sharing your wisdom and knowledge. It's great to have you with us.
Pleasure to be here. I always like to be connected with Australia because there is so much going on there.
Well, look, as an Australian, we love to hear that with people wanting to be connected with us, because we tend to look overseas at people doing amazing things and, you know, put them up on a bit of a pedestal and say, we need to be more like that. So I am hoping that we get some of that inspiration from you here today as well. But tell us about Earli and what you're doing. I mean, going from working in consumer products to the cancer space is a massive change and really different. Why and what are you doing?
I made a decision to follow my heart and not my mind. After the last company, Shopkick, which was sold and acquired, it was one of those moments where I was planning on taking 2 years off. It was a hard road, but it worked out really nicely, and the company was sold for $250 million. And I wanted to take time off, and I initially had a lot of fun. I went to China and looked at the Chinese startup system and the Chinese entrepreneurs, spent time in Beijing. We picked up our little micro children. At the time, they were 3 and 1, and went to Beijing for 3 weeks and then to Shenzhen. And it was a great time. And then we went to Europe, and that was the first 4 months, and I really loved it. And then after 6 months, I became very unhappy, because I felt very useless. I was not doing anything except emptying the dishwasher at home. And that's useful, but I can do that also with a job. So I was basically saying something needs to change, and I think I'm not going to retire anymore from now on. So I made a decision to follow my heart and not my mind. My mind would have brought me back into some mobile commerce thing. My last company was called Shopkick, which is a shopping app that rewards you simply for walking into a store without buying anything there with your smartphone. And that worked out all really well, but my heart told me something else. I was following what I was really interested in. And I observed what I was reading, what I was talking about, what I wanted to know about. And it led me to science, because I love science. I'm not a scientist, but I love science. So I looked at everything, and I ended up looking at very different things, very strange things that my heart brought me to. For example, brain to machine interfaces. I was literally helping a guy sitting in a dilapidated building in Berkeley, shooting microwaves at his own head, trying to decipher his own thoughts.
Crazy.
It was really interesting, but a little too early. And then I looked at consumer robots, and I believe everyone's going to have a robot in their house in 20 years from now. It's going to be very strange not to have one. But it's also a little early. And then I looked at software meets biology and engineering. And on that intersection, I became very, very excited and interested. And when you think about biology meets engineering and software, this world's colliding, the wet lab with the dry lab, you very quickly talk about precision health, which is the idea that different people need different forms of treatment or interventions or preventions. And since I only live 4 miles from Stanford, literally, in a small town called Portola Valley, I decided to roll down the hill and go to Stanford, and I stumbled around the campus, and I talked to all these very exciting and interesting people, probably 100 of them, and each one was telling me that we're working on the world's most important idea.
Yeah.
By definition, 99 of 100 were wrong, likely all of them. But because I'm not a biologist, I had a very hard time differentiating a good idea from a very good one from an outstanding one. I was probably able to distinguish the bad ones and from the good ones, but not whether it's good, very good or outstanding. Only outstanding ideas are worth pursuing. And they're very rare. And good ideas, there's an ocean of them. And it's really tempting to fall into the trap to say, well, this idea is pretty good. I'm going to do that.
Yeah.
I didn't want to do that at all. In fact, my very first filter when I hear something new is I try to forget it.
Right. Okay.
And only if I cannot forget it, despite trying intensely, it might be potentially interesting. That's just the first filter.
I love that filter.
So then fast forward a few months, I still hadn't found it. And I was getting quite frustrated. It was Thanksgiving Day of 2016. And when Thanksgiving rolls around, for me, kind of like the year is kind of over. And I felt like I hadn't accomplished anything because I still didn't know what to do. So then my wife, Angel, comes into the room, and she holds a physical print magazine in her hand, of all things. And the reason we got this magazine was because we lived in somebody else's house, because our own house was being remodeled. So it wasn't even our magazine. So she comes with this magazine. She says, this is from Stanford Medical School. There's this article about precision health. You may want to see that, because she knew I was looking into precision health. And I said, okay. So I looked at the magazine, and it was an article called "And Yet You Try." And this article was about Sam Gambier, one of the world's most preeminent early cancer detection experts. He was at Stanford, and how he lost his only child to cancer at age 16. I read the story, and I was shaken by it. And I decided to send Sam an email that day, a cold email. And I said, it's not a coincidence I'm sending you this email on Thanksgiving Day. I can only imagine how hard this day is for you. It's a holiday. I don't even know your son, but I felt a gripping sense of grief. But I was also strangely inspired by the work you're doing. I am not a scientist. I'm not a biologist. I'm a serial entrepreneur. I'm looking to build my next thing. I'd like to meet you.
Wow.
2 months later, we met for breakfast on a Saturday morning in Portola Valley, 4,000 people. It turns out he also lived in Portola Valley. We just didn't know each other. We were neighbors.
Wow.
We spent 3 hours together that morning, and you asked why somebody with no background comes into this world. And I actually started the conversation by asking him that question. And I promise you, I don't like humble bragging. So this is not a humble brag question. This is a real question, because the last thing I want to do is waste 10 years of his or my time or anybody else's. So I asked him the question, should somebody with my background, or better, lack thereof, bother the world of biology with my presence? And he said, I'll give you 2 answers. The first one is, I believe the world of biology needs more people like you, not fewer people like you, because we have a lot of experts and very few generalists, and we have even fewer people who know how to build startups. Yeah. The second answer is, the world of biology will always find a way to screw you over. It will take longer than you want, and it will be harder than you want. So I didn't particularly like the second part of the answer, but I took it as a cost of doing business. Yeah. And then before I knew it, every other Saturday morning, I would find myself sitting at his kitchen table, and he was trying to teach me biology, largely in vain, but I give him credit for trying. And then he connected me with a bunch of people at Stanford in order to help me find my next thing. And 3 months went by, and I came back to Sam, and I said, I'm so sorry, Sam, I still don't have it. It's not the right combination of idea and people. May I ask you a simple question? You've seen hundreds of ideas in your own lab and outside of your own lab, probably just in the last couple of years alone. Which one has the highest potential of all? And he looked at me in his scientist underselling type of way. And he said, there is this one idea that I believe has some real potential.
Yeah.
And that became Earli.
I'm having a moment that is, it's really rare for me. I hear a lot of founder stories, right? I hear a lot of people of how they had their why and how they had their aha and how they've come to their, this is going to be your life's work, not the sum total of it, but a really big part of it. But hearing your story and the humanity in it and the connection that you two have built it's quite profound, actually, I think, to as a driver, as a motivational driver of thinking about how you go forward. So I just personally, thank you for sharing that. That actually resonates so strongly with me. That's awesome. You've got the idea now. How do you go on to raise, a Series A of $40 million and have clinical trials in Australia? I'm leaping forward. I haven't even told people what you're doing, or we haven't even talked about what Earli does. But, you've gone from that journey of this cold email, being so personally moved, and sending that cold email saying, I think I need to connect with you, and recognizing in that connection and that real human relationship, exploring an opportunity and a problem space, to doing, to where you are today. What's that journey kind of been like, if you can kind of pull through some of it? Because it's a massive leap, right? And it's not everybody has that moment so deeply that you've had.
Well, it's a very intense and gripping journey. Many things happened since. The idea of Earli came from Sam and his lab, and a talented postdoc working in his lab. And they were turning things on its head and in a way that was so profoundly different. Maybe I should briefly say what we do.
Yeah.
So basically, the question is this. There are all these people that are trying to find cancer earlier, because finding cancer earlier is the single biggest lever to more survival. It's better than any therapy, right? Yeah. You can drive up the 5-year survival rates of the 5 biggest cancers by 4x up to 12x, depending on the type of cancer. Can you believe that? I mean, imagine the numbers. We're talking about millions of people a year.
Yeah.
So early detection is important, and a lot of other people have been working on it by taking blood samples and looking for fragments of cancer DNA or methylation patterns in blood samples. It's a beautiful idea. It's very elegant. In theory. In practice, unfortunately, is extraordinarily hard, because if you take an 8 milliliter sample out of your body, out of a 5 liter total blood volume, there may not be anything in the sample to begin with.
Yeah.
And if it's really early cancer, you can imagine how small that is. And there's very little material being shed. Some cancers hardly shed anything. So then you can't find anything. And it doesn't matter how good your analysis afterwards of the sample is if nothing is in there to begin with.
Yeah.
So a company called Greylock, which is the biggest player in this liquid biopsy space, spent $2 billion with a capital B, not an M, on R&D. And the figures they published were 42% sensitivity for stage 2 cancers and 19% for stage 1. That is after 2 billion, and it's for the 20 cancers they selected. It doesn't include lung cancer or breast cancer, the 2 biggest killers. So what are we going to do about this problem if it's so hard to do? And there's yet another problem. For some cancers, it might work very well, like colon cancer. Pancreatic cancer looks promising, and I'm rooting for them because anything helps. Imagine the opposite scenario. Imagine you find every little cancer that exists. Now you have another problem. The cancer may be so small you can't image it anymore. If you can't image a tumor, what are you going to do about it?
Yeah.
The clinician, and you have clinicians in the room, they're not going to give people chemotherapy for a lesion they can't even see.
Yeah.
So now what are you left with except sending people home to tell them they have cancer and now to worry about it, but they can't do anything about it. And that's kind of defeats the purpose of early detection, doesn't it?
Yeah.
So that's how Earli was born, out of this frustration, because Sam was sitting on the advisory boards of various of these companies. And he asked a very profound question. What if we stopped searching for cancer altogether? What if instead we forced the cancer to reveal itself? What if the cancer was forced to make itself known and even help destruct itself? So that's where Earli comes in, because the idea of Earli is to make early cancer detection actionable and help with its discovery, but also help localize it and perhaps even treat it. And this is how it works. Imagine you could get an injection and it disperses a compound that is going through your body and randomly enter cells, healthy cells, as well as cancer cells if there are any. But only if it's a cancer cell. It will force the cancer to make a non-human synthetic biomarker that doesn't belong in your body, such as limonene. Limonene is in oranges, not in humans, unless you eat them. If you exhale limonene after getting our injection, you might have cancer because the cancer was forced to make limonene. Now, suspend disbelief for a moment and imagine this all worked. You still haven't solved the problem, because now you might know there is cancer somewhere, but you can't tell me where it is. Same problem as with liquid biopsies that work. Then what are you going to do about it? So now step 2 kicks in. This time we're forcing the cancer to make an enzyme that eats up a radioactive PET tracer that has been injected, and it traps the tracer inside the tumor that's normally too small to image, and makes that tumor imageable. And by making it imageable, it makes it localizable. By making it localizable, it makes it treatable. Because now we can use precision radiation or targeted surgery to take it out. And in the future, you may even be able to use the Earli platform itself to force the tumor to make its own toxin and kill itself, or to activate the immune system. So the uniqueness of Earli lies in the fact that we're introducing an artificial construct into the body that disperses throughout the body, and it enters healthy cells, benign tumor cells, and cancer cells. But only if it's cancer, it forces it to make an enzyme that traps the tracer and makes it visible. And the trick is we're not using chemistry like everyone else is. We're using biology.
Yeah.
And biology has the advantage that it allows for massive signal amplification.
Okay.
And Sam Gambier, when we started the company, said, we cannot rely on biomarkers that nature may or may not always produce for us.
Mm.
But if we engineer the system, in other words, if we control it, we can produce consistent signals that predict that there's cancer. We call that a synthetic biopsy, a new category that hasn't existed before. And that was the start of Earli, when we used that, took that from Stanford and licensed that exclusively into the company. And then we had nothing except a license. And then took me 9 months to find my co-founder, because I'm the clueless guy in the room. I don't know anything about biology, so I'm kind of worthless. So I was looking for the person who actually knew their stuff.
Yeah.
And I had a very tight filter. I was looking for somebody with small company, large company, public, private experience, and gene therapy experience up to phase 2 trials, very few phase 2 trials in the world with gene therapy.
Yeah.
And then I have a personal no asshole rule.
Yeah.
And all of that, as you can imagine, makes the Venn diagram quite small. And then I got very lucky. I looked at over 200 people. I found David Sui, and he joined as our chief scientific officer and co-founder. And Sam was on the board, and we started the company. And then we took that whole thing, including the license from Stanford, to Mark Benioff, the founder of Salesforce, who I know. And he said, oh, this is awesome. I want to be part of that. And then we met with Andreessen Horowitz, one of the biggest venture capitalists here in Silicon Valley. And I knew Mark Andreessen from before. I met him and I said, hey, I got this thing going, which is kind of crazy. I think you may want to see this. And before we knew it, we had raised $19.5 million in a seed round and got going. And then we got lucky and got into Johnson & Johnson's incubator called J-Labs in South San Francisco. And then we started hiring our first people, and that's how we got started. And then for the Series A that you alluded to, the 40 million, that was very difficult because what happened in the meantime was an extraordinarily sad event. Sam Gambier, who I was very, very friendly and not just, he's just an incredible individual. He passed away from cancer himself. And it was the worst because it was a cancer of unknown primary. So there was no way to detect it early. And by definition, it was stage 4 when it was detected. It was an incredible loss for the world, for his family, and also for us and for me personally. I was very, I didn't know him that long, only a few years, but I was extremely close with him. So that happened. And then it was COVID and we only had everything, all video, every pitch was by video to investors. And we had been in semi-stealth because we wanted to produce new data before we go out to the market with what we've done. Otherwise, it would just be that Stanford company. And then we only had mouse data at the time. So that was our pitch, and we wanted to raise $40 million in the middle of the COVID lockdowns. And then we needed to find a solution. So I'm going to pause here for a moment.
It's quite incredible. Like every milestone in your journey, or every pivotal moment, which are not necessarily milestones of the company's progression, really has unlocked something new, either in a way of thinking or the next step, which I think is quite amazing to hear. Even for something so for something that seems so fantastical, seems so unreal, seems so outside of the box and outside of the way we're thinking, to get to that point of raising that first seed round at such a significant amount of money. At least for Australia, we don't see seed rounds in the biotech space yet like that. I hope to one day. But to get to that point and bring people on that journey, I think really speaks to a couple of things that at least is what I'm taking away from this is one, that this is actually, yes, it might not be the typical pathway that others have approached, the early stage detection process, but it is actually founded in science, and there's a really committed team, and you're creating and curating an amazing team who are willing to go on the journey and work at this, really systemically to solve the problem. But the second one is, I think that, you've spoken about so many people being touched by cancer, and I think every single one of us has been touched by cancer, either personally or with our family or with our friends along that journey. So there's a desire, and I think an appetite for a solution in this space, or for solutions in this space, because people have been impacted personally, like yourself, like me. I shared with you earlier about my own personal journey with my family right now. And we want the hope, and we want people to be bold and ambitious and to try the really hard things to help, because we need more hope in the world. And quite frankly, after the last 2.5 years with COVID, we definitely need more hope in the world around these things. So you've pitched, you've raised money. I don't want to rush your story or leap ahead too far, but I do, this is impressively fast. Most biotech companies, or most sort of, therapeutics or diagnostic companies, it's 5, 10, 15 years. In this time frame, we're talking about a 3-year window in that time frame, aren't we? Roughly so far, 3 or 4 years?
The company was founded in June of 2018. So we're now 4 years old. And wow. We were talking to our investors, David and Sam and I, we were in sync on this, that we wanted to not fall in what we call, let me first say, we went to our investors, A16Z, and we told them we want to be in humans in 3 years from now. Yeah. And they kind of kindly chuckled. They're rooting for us, but they were basically saying, well, that's great. Good luck. They had obviously heard this before.
Yeah.
So then we sat down and we said, okay, why don't we just reverse engineer this a little bit? So let's say we have to be in humans in 3 years from today. What is going to keep us from doing that? So then we sat down, and it turns out it's 3 things that would keep us from doing that. The first one is what we call the mousetrap. There's this old joke about biotechs. You go to a biotech startup and you ask them, when are you going to be in humans? And the answer will be in 2 years from now. And then 2 years later, you go back, you ask, when will you be in humans? And the answer will be in 2 years from now. So and the reason why is because they keep testing in mice, and there's always something to improve.
Yeah.
So you're never really done, and you just got to do a little more until you can then take it to humans, right? But that gets you trapped in the mouse trap. So you got to get out of the trap and at some point make a decision. This is for now good enough to take to the clinic. We're going to test this.
Yeah.
And yes, we know that by the time we inject our first human patient, we already know this is not good enough, and we have something way better at the time. But that will always be the case. So you got to leap forward. Yeah. Secondly, the FDA approval cycle for a new phase 1 might be somewhere between 12 to 24 months. Well, that takes 1 to 2 out of your 3 years away right there. That's not very helpful. So then we traveled to China and we looked at that system. Turns out that's not very helpful. Then we went to the Cayman Islands, and actually we didn't go there. I would have loved to go, but we didn't. But it turns out they don't have enough people living there. And then we looked at the Netherlands, and that's really, really interesting. But then we looked at Australia, and that's even better. So why is Australia better? It's because you have a system of clinical trial approval that happens at the local level, not at the federal government level. Through ethics committees. And that shaves 6 to 12 months off your timeline because it's much faster. And yet the quality of the clinical trials is extremely high, so high that the FDA routinely accepts the data coming out of Australia if you run it the right way. And in Australia, you've got highly capable, experienced and talented physicians, clinicians, and a clinical system that's absolutely a top notch, and you've got 15 million people living in condensed areas of the largest cities in Australia that are ethnically diverse. So this is extremely interesting. These trials are well run. Everything is highly professional. So this is a great place. The only problem is it's pretty far away from where we are.
Just a little.
Just a little. But otherwise, it is perfect. So we decided to come to Australia. We traveled there in early 2019. And the good news was that David Suey used to work for an Australian company before. So he knew several people, including one wonderful person in Australia that we worked with who was going to manage our trial. And so we formed a 100% owned subsidiary in Australia called Earli Pty Limited, which is designed to run the trial. And then we got very lucky and signed up incredible partners with Olivia Newton-John Cancer Center, Andrew Scott, and Huey at Olivia Newton-John. And then we signed up Michael Boyer at the Chris O'Brien Lifehouse in Sydney. And then most recently, we added 2 more clinics, Icon in Adelaide and Paso in Frankston. And these 4 clinics are now our partners in Australia. And then, of course, we had to, the third big thing is what slows you down is GMP manufacturing of the product that you've got to get into humans. So we formed a special partnership with Aldevron, one of the partners here, to accelerate the process of manufacturing. And then we went through the approval cycle, and on June 27th of 2021, we dosed our first human patient in Australia. That was 3 years and 2 weeks after we founded the company.
Can I just say how phenomenal that is? And I'm literally going to write this up later and be like, when people tell us that can't be done, I'm like, no, it can be done. It's just really hard, but it can be done because we've now got a case. We've got a case study. Congratulations. That's a massive milestone. guessing it's way too early to tell us much about how the trials are going, but is there anything that you can sort of say? Are they progressing well? Are you happy with how things are going? When do you think the next, there might be something to sort of share with us, and what does that process look like for you?
It's going well, but we can't disclose anything yet. It's too early, and we still have to go further. We still need more patients. Right. We've been hampered with our enrollment speed. We're not happy about the enrollment at all because it happened during COVID lockdowns. And obviously Australia took COVID a little more seriously than America and shut down its cities. I think Melbourne was one of the most locked down city for several months of the world. That didn't help particularly with enrolling into patients into a clinical trial. So you can help us by spreading the word about the trial, by letting people know about it, because we need more patients to join. These are late stage lung cancer patients that we're enrolling, stage 3 and 4, and we're starting with late stage because that's the way you start testing something new. And then we're going to move to earlier stage over time. So that will be our focus, getting more enrollment in Australia. Anything that is helps that makes the trial more known. And you can easily contact the clinics, Olivia Newton-John Cancer Center, Andrew Scott there, or Michael Boyer at Chris O'Brien Lifehouse. You can also send an email to Earli at trial@earli.com for more information. So there are ways to reach us. Now, once that trial produces data, then we can go to the next step on the human side. In parallel to all of this, we're working on the next generation of our preclinical compound, because we're now moving into localization. Not just saying there is cancer or there's not cancer, but actually, where is it? Amazing. Where is it in the lung? And the goal is to light up malignant nodules and not light up benign nodules. And to give you a glimpse, in lung cancer, 94% of all nodules, these are these little lesions in the lung that you see on a low-dose CT scan, are benign. Only 6% are malignant. The problem is no one knows which 6%.
Right. And
so the standard of care is to send people home and ask them to come back 6 to 12 months later to see if anything has grown in the meantime. And that's called, euphemistically, watchful waiting, which I think is like a terrible expression. Because in that time frame, your very small homogeneous tumor with very few mutations might have turned into a heterogeneous tumor with many mutations. And then what happens is you see this often, people get chemotherapy and they go into remission and everybody's celebrating. Unfortunately, that chemo kills 48 out of 50 mutations, but the 2 mutations it can kill will kill you. So checking this early and finding it early is critical in order to have as few mutations as possible.
I love that you're doing clinical trials and you're already working on the next thing, right? That's phenomenal to me. And we will actually make sure that we go out and we share with our community how to get involved, because I would love to see more patients go through. As I say, I think everybody in the world has been touched by, or if you haven't been, you will be touched by cancer, unfortunately, in your circles at some point. And so we have a vested interest in actually collaborating with you and driving people towards this for you to be successful, actually, at the end of the day, and for Earli to be successful. There's something I want to unpack there, because I've heard you speak about this. I've read a few things where you talk about Earli being a bioengineering firm versus a biotech firm. And I think some of the lessons of how you're approaching where you want to be and that rapid pace that you're able to get to clinical trial. Can you unpack a few of those lessons of the difference in the approach and how that software engineering and that sort of, massive scale growth and adoption thing, how you actually are bringing that to the biotech space and really engineering your company for the outcomes that you're driving towards? Because I think it's really quite a valuable mind shift for some of our clinical folks out there to hear.
It's a very good question, Sally. It's a very different approach. Essentially, think of it as the difference between serial and parallel work. The idea is to iterate your way forward like an engineer, where you try something And while you're trying it, you try something else, you break it into pieces. And you have multiple things happening at the same time. In our case, it's 4 pieces, and we cut it into 4 teams, and each team is iterating. And then the trick is to bring them all together. I always have this image in my mind of sailboats that are in a regatta, and they're all going together. And they're, instead of racing and trying to win, they all actually have to come in at the same time towards the goal and have to reconvene, which is very risky and very difficult, but it's extremely efficient if it works. So that's the parallelization piece. The other part is the iteration piece, where instead of taking the typical pharma approach is like you try to go for a small molecule that's going to cure cancer, hopefully, and you spend $1 billion, and then you find out if it works or not, right? And there's many years. And I admire those companies for doing that. And somebody has to do it. I would never do it. I'm a risk taker, but by nature, but that, right? Like some of the people that we bring on board who work at Earli come from a large pharma background, and they are routinely shocked by our approach because we're basically have to explain to them. For us, it's very natural because we live this world. We live this, right? Yeah. But for them, it's unheard of to run a phase 1 trial on a product that we're not intending to bring to market. We're intentionally never intending to bring the current product in Australia to market. What we're trying to prove is that the basic concept works in humans. And if the basic concept works in humans, then we have a very good shot that our better product is going to work even better in humans. But if we are not working in humans, we had better fail fast. So we need to find that out very quickly to know if there is a fundamental problem with the construct or not. So that's why we're doing this. Now, we're not going to run 5 or 6 phase 1 trials just for the sake of iteration. We're not going to do that. That's too costly and takes too much time. But the whole approach at Earli is different. For example, our intermediate model between mice and humans is dogs. Well, that's a road less taken too. Because dogs actually are very interesting for us because they are a large body mass. They're like 1,000 times larger body mass than humans, than mice, but only 2 to 9x smaller than humans, depending on the type of breed. So now, large body mass, and greatly, they are sort of like humans in the sense that they are developing, unfortunately, I should say, spontaneous cancers. In fact, more frequently than humans. So there are 6 million dogs every year in the US that develop cancer, and they are taken to the vet in order to help them. And those become our patients for our clinical trials with dogs. So that helps us accelerate the whole process of not going into humans every time.
It's quite remarkable and methodical. And I think, again, I feel like there's a whole lecture series on that we could unpack for some people to be sort of like, here's some lessons from software that you could apply and how to think differently. But I think it almost goes full circle to the start of your story with that very first conversation and the sort of question that you posed about whether somebody like you should go into the field of biology and be part of something like this. And the answer being yes, because you have, you're coming from a different and a diverse background and a different perspective. And it's really powerful. I'm conscious that we don't have too much longer with you, but I've got a couple of quick questions and then I want to, give you the floor to anything. But I think the 2 quick questions that I wanted to ask was first of all is you did mention people that come from pharma and work for you. So how do you address culture and in the organization? Because while this is a scientific company and, science is the heart of what you do, people are the people that are doing that discovery work. So how do you think about culture? And you sort of said you have a no assholes rule for founders. I assume that applies to employees a bit as well. So I'd love to hear you a little bit about culture. And the second part is also a culture question, which is also, you know, in Australia, we see this quite often in universities. We see this really focus in that research needs to be pure and it's over here and commercialization is over here. But what you're actually talking about with Earli is doing both commercialization and some fundamental research, because I'm imagining there's lots of IP being created and new things being done, not just in a process perspective, but in what you're using and a lot of discovery work. You're actually overlapping them, which is something I think we need to see more of. So I'd love to just get your perspective on culture inside of the organization, and how do you think we create that culture where commercialization and research can be a little bit more hand in hand? So two light and easy questions.
I'll start with the second question, because I think it's a great dovetail into the answer for the first. The commercialization of a product in healthcare is the only way to help patients live longer. So if your goal is to actually help people, you're gonna have to commercialize.
Yes.
Sooner than later. So that means if you believe that commercialization is the bad thing here, I think you might have fundamentally misunderstood of what it takes to get this into patients, because there is no patient benefit unless there's commercialization. So these are not exclusive and these are not counterproductive if you have the mindset of helping patients. We are only here for one simple reason at Earli, is to extend people's lives, period. Everything else is a side effect. And if that is not our first goal, our North Star, we are not understanding what we're here for. So I give you an example. We had our very first patient who went through the Earli trial at the Olivia Newton-John Cancer Center. Ted Cunningham joined our virtual all-hands meeting at Earli and addressed the whole team. Can you imagine what this means to a scientist who is standing in the lab, formulating our compound and trying to find the next iteration of it, to hear from a patient that actually went through using it? Can you imagine what that would mean to somebody at the university as an academic? Most ideas never see the light of day. We are in the business of making ideas see the light of day so that they can help patients. So that brings me to culture. Our culture is about patients. It is about values that lead us to that goal. And any milestones along the way, like financing, millions of dollars, raising money, having a nice lab, they're just means to an end and nothing else. So the first big milestone, the first really, really big milestone is not the next financing round or an IPO or anything else. It is when the first patient's life gets extended. So in terms of culture, that means you got to find people who are mission driven, who are not there as a mercenary, but as a missionary, who are there because the mission matters. And our mission statement is kind of crazy sounding, but maybe now that you understand what we do, it might sound less crazy. It is to make cancer a benign experience.
I love it.
Because if you find it early and you can localize it early, then you might be able to do something about it and turn this from one bad chapter in your life from your last chapter in your life to one single bad chapter in your life and keep living on. And that's what we do at Earli. So our culture needs to reflect that. So we have values that are very important for us, like integrity and honesty are at the top and nothing else until a long time after. Integrity, I don't know what your definition is. Our definition is you do what you say and you say what you do, which means actions and words are congruent. They're the same. So what I tell you is actually what I'm going to do. And what I'm doing, I'm going to talk about and not wipe it under the rug. And combine that, you can have, by the way, you can have perfect integrity and be a liar like Donald Trump, where I'm going to go on the record with that. I have no shame on that. So where you tell lies all day long and then you actually act on them, that gives you perfect integrity by definition, right? So you got to couple that with honesty, which is telling the truth.
Yeah.
If you tell the truth and you have integrity, that's an extraordinarily powerful combination. It's also very hard to sustain every day, by the way. It's really hard work. You constantly do what you say and say what you do, which means you speak about what you do and you act on what you tell people. If I'm going to send you this thing on Monday, I have to send it on Monday, not on Tuesday. And if I can't get around to it, I have to say, I'm sorry for that. I didn't make my time, but I'm going to make up to you. Tomorrow at noon, you will have it, and then stick to that commitment. So those are the little things. And of course, there are much bigger things about transparency and how you share information with the team. And then work hard, play hard, which means not just be the party gang, but it means enjoying the journey, which the journey is really hard. So we had better enjoy it, right? In fact, perhaps we're partially enjoying it because it's hard. It's like why do people go up the Mount Everest? Not because it's easy. So you're doing things because they are difficult and they matter. And the combination of difficult and matter can only be accomplished with amazing people. And you can only attract amazing people with amazing missions and great values. So that's our culture.
So I'm pretty sure that anybody watching this in the future or pulling out from this is going to be a little bit in the same state as I am. As I'm just blown away. I rarely am so inspired by the work somebody's doing that I sit there and go, I'd like to come and work for you. But I'm sitting here right now just in full, I guess a little bit in awe, but also just really, really thrilled to hear that articulation of values and being a values-led business and a values-led company that the whole mission, just everything you've articulated then sings to my heart, really. So I might be fangirling a little bit, but I just think it's such important work and the articulation of your purpose and your why, but also how you get there, and not just from the engineering and the science perspective, but how as a team and what that means is incredibly important to hear. And I think it's something we don't often hear enough of, particularly if I dare say it, having worked in big tech in Silicon Valley. We hear values used a lot, but lived out as you describe it, and that integrity, it is hard to live out your values, and they should be hard to live out because nothing easy is nothing that's really difficult to achieve is easy. It requires that everyday work. So I think it's just absolutely incredible. And I think the work you're doing is incredibly inspiring and incredibly important. If we wrap up on sort of one final question, what is it that you would like anybody that is watching this, that is engaging with Earli or is hearing from you, what is one thing that you would like people to do other than, refer people to the clinical trials? Because we'll definitely make sure that we do that. But what's one thing that you'd like people to take away from this? Because I think it's rare for us to get an opportunity to hear from somebody as committed and passionate and driven as what you are. So there's this kind of, what's our life learning moment from you that you kind of call to action that you want people to take?
Follow your heart, not your mind.
Wow. Cyriac, thank you so much for today. It's been, I cannot tell you how much I've enjoyed this conversation. For me, it's rare to walk away. I'm around amazing people and founders every day, but sometimes in the grind of it, you miss that moment of inspiration for yourself. But I'm walking away quite inspired for myself and Cicada and what we do here to actually go do the hard stuff today and not do the easy stuff.
You're doing what you're doing because this is great for Australia. It's great for Sydney, and it's great for the world. It's needed. The more you can get founders to engage in working on the hard topics, on the important topics, the better for everybody. So I'm a big believer in what you do, and I appreciate you doing that.
Thank you so much. Look, thank you for your generosity of time and your wisdom. When you are down here next, we would love to host you at Cicada and get you involved and celebrate and hear more about what you're doing. It's been an absolute pleasure. For everybody watching, thank you so much for joining us. This event has been part of the Visiting Entrepreneurs Program from the City of Sydney, and we're delighted to see that support from the City of Sydney, from Tech Central, from the Greater Sydney's Commission to connect us with amazing people around the world, to inspire both the people inside of Sydney, but more broadly across Australia, to actually follow their heart and do the hard things. Thanks, everybody. Bye for now.